This presentation deals with the use of ECIS to monitor signal transduction in cells in culture. Before looking at the beta-adrenergic receptor, let's consider the response of cells to the second messenger, cyclic AMP. Here we're looking at dose-response curves where MDCK cells have been exposed to a membrane-permeable cyclic AMP analog, CPT cyclic AMP. Notice there is a strong change in the impedance that is a function of the dose of this compound added to the cells. Again, this is a membrane-permeable cyclic AMP. In this slide, we see results when the beta-adrenergic receptor is activated on cells using the agonist isoproterenol. Notice when this compound is added at a micromolar concentration about 5 minutes into this run, the confluent layer of cells on the electrode goes through a striking increase in the impedance due to a morphological change elicited by the signal transduction cascade. To show more thoroughly that this is signal transduction, we bring in an antagonist, ALP. In this particular trace shown in panel A, 5 minutes into this run, the confluent layer of cells is first exposed to 10 micromolar ALP, and then later, 25 minutes later, 1 micromolar of the isoproterenol is added, the agonist. And notice the response, which we see in panel B without the antagonist. The response is lost in the presence of the antagonist. Yet later on, at 65 minutes in this experiment, 2.5 micromolar forskolin is added to boost the cyclic AMP second messenger, and the response is regenerated, clearly showing that this is signal transduction that we're detecting using impedance measurement. Now let's look at signal transduction using the ECIS model. This model is described more thoroughly in one of our presentations dealing with ECIS theory. Here we're going to use Chinese hamster ovary cells that have been engineered to overexpress the muscarinic receptor. The agonist in this case is carbachol. The Chinese hamster ovary cells are grown to confluence and then exposed in the control just to buffer or in the other traces to different concentrations of the agonist carbachol. Looking at the response of the cells, it's quite striking. The resistive portion of the impedance measured at 4,000 Hertz has gone from about 5 kiloohms up to 10 kiloohms, a nearly doubling of the impedance. These curves show the proper EC50 value of approximately a micromolar, again suggesting that we are seeing signal transduction upon the addition of the agonist. To more thoroughly demonstrate that this is signal transduction, again we bring in the antagonist perenzapine. Here again the CHO cells have been grown to confluence. But we're looking at the normalized resistance. In other words, all the resistances have been divided by their starting value, so all of these traces start at 1. Again, we add here perenzapine at different concentrations indicated on each curve, and 15 minutes later we add the agonist carbachol at 20 micromolar. Notice the magenta trace with no perenzapine added shows again the striking response elicited by the agonist, a strong increase in the resistive portion of the impedance. But as we add the perenzapine, we can clearly attenuate this response, and it almost is completely eliminated at 0.5 micromolar perenzapine, again strongly suggesting that this is signal transduction. When we actually look at this confluent layer of CHO cells, we cannot discern in a microscope any difference between those that receive the carbachol and are stimulated and those that are the control So what is taking place to cause this striking increase in the resistive portion of the impedance? Again, we are not going to describe the model in detail in this particular presentation, but the fact that current flows through different paths depending upon the frequency, the AC frequency of the signal used to make the measurement, allows us to take apart different parameters in the cell using the model. Specifically, the model can separate out the problem of current flow going beneath the cell, a term that we describe as alpha, from the problem of current going between the cells through the paracellular space or the barrier function of the cell layer. Because measurements made at high frequency couple directly through the cell membrane, the model also can return data related to the membrane capacitance of the cell. Now let's look at the raw data first on the left-hand side of this figure. Here we're looking at the response of the CHO cells to 100 micromolar carbachol. Again, a strong increase in the impedance. The panel on the right, however, this raw data, has been broken down into three parameters, namely RB, the barrier function of the cell layer, shown in green, alpha, the parameter associated with current flow in the narrow space between the electrode and the basal membrane, and finally, the curve in blue, the membrane capacitance. What is clear from this analysis is that the increase in impedance seen upon the addition of the agonist carbachol is almost entirely due to an increase in the barrier function of the cell layer. Presumably, as the cell, something in the signal transduction cascade has resulted in a tightening of the cell-cell junctions. Again, this shows the power of the ECIS method by making measurements at multiple AC frequencies. Thank you for joining us for this brief presentation dealing with the use of ECIS to measure signal transduction.